Hi guys, welcome back to Biology Videos at TDA. This is the Year 12 and 13 lesson, OCR 12.7 on preventing disease. To start us off, let's see if we can guess who this gentleman is, quite an important person. So we've got Alexander Fleming, so he was one of the first people to discover antibiotics and help us learn about actually using them. Okay, so for today's lesson, we're going to do a bit of a recap and then we're going to be looking at the differences in passive and active immunity and then the differences between natural and artificial and the combinations of. We're going to be looking at vaccination in a bit more detail and the uses of it, and then how we manage the use of antibiotics in sort of modern society. So as a recap, some questions from last lesson and a couple that kind of move on to this lesson at the end. So if you pause the video, have a go at these. Okay, welcome back. So we're going to look at now then the answers. So what do we call molecules which mark antigens? So we call them opsonins, mark, tag, same thing. What do we call these Y-shaped glycoproteins? Well, they're our antibodies that we looked at in a bit of detail yesterday. 4 ways in which antibodies defend the body. I repeat this quite a lot because I think it's quite important to learn. So first one, opsonins, they can tag them. Second one, they can make this antigen-antibody complex that we learned yesterday. Makes it less dangerous. They can act as agglutinins. And then finally that clumps them together, stops them spreading, easy to do phagocytosis, and finally they can be antitoxins. Question 4, where do they mature? So the B cells mature in the bone marrow, easy to remember. The T cells mature in the thymus. Just to clarify from yesterday, if you hadn't seen the update, the T cells, they are made still in the bone marrow, but they mature in the thymus, so just to clarify that. And then number 5, 3 common autoimmune diseases. So these are diseases caused by the body. You don't catch them. They're not transferable. So type 1 diabetes, rheumatoid arthritis and lupus. Just some examples. Okay, so we can look at the actual types of immunity now. So the first one we're going to look at is natural passive. And the words themselves are really useful in terms of kind of learning them. So if you can understand the word itself, you can then understand the type of immunity. So natural, the idea it's not man-made, it's done by living organisms themselves. And then passive, that means to essentially get it from someone else. So it's naturally made, but then you're getting it from someone else. And the only real example I have here is the idea of the mother's milk. And the very first milk that mothers make is called this colostrum, this kind of yellowy looking, not very nice looking milk. And in there's many antibodies because when a baby's born, their immune system isn't as well developed. So they'll need the antibodies. And they'll have the antibodies from the mother that will be the likely pathogens in that kind of environment, and that should give them enough immunity until their body develops themselves. Okay, and then let's move on to natural again now, but this time active immunity. So this is quite simply getting a pathogen and becoming resistant to it. In sort of modern society, we take a pill or a drug for pretty much everything. Something goes wrong, we think we need a pill to cure us, but we don't. Most of the time, most of the illnesses we're going to get, your body's going to fight it off themselves in the ways that we looked last lesson, using the T and the B cells, and they're going to destroy it. So this is active and it's natural, so it's just being done by the organism itself, and it's active, so your body's having to make the antibodies, they're having to go through a process to respond to it. And really common one which we look at is Campylobacter. You've probably had it if you've ever had really bad sickness and diarrhea. Common bacterial infection is the Campylobacter. You don't need antibiotics to fight it off most of the time. Some people do, but most people won't need to. They can simply fight it off themselves. So we call this natural because your body's doing it itself, and then active, so you're having to actively produce the antibodies in what we learned yesterday. Okay, so that takes us on now then to artificial active, so the most common kind of way that we learn about in terms of vaccination. So what is a vaccine, first of all? So I've tried to really simplify vaccination for you because you should have a reasonable idea now. You should have had many vaccinations in your life. So we can take a pathogen and we can simply inject it, or we can give a dead version of it, or more common versions are attenuated, weakened pathogens, or toxoids, so they're sort of made harmless. And there's various other ways you can gently modify directly of antigens themselves. And your body can get immunity from that. So your body will then go ahead and do the primary response, similar to the natural active. Your body will then respond, but this time it's artificial because you're being injected with a pathogen on purpose. Now, looking at the current UK government, this kind of herd vaccination or herd immunity is what it's being referred to. as we're trying to fight off the coronavirus. And the idea here is if you have one person who is immune to something, and then more and more people that are immune, it means you're less likely to then suffer from it. If I can do a really bad, let me see if I can draw some people. So these are my little people in an area, and the idea here is, and all these people here in red on the outside, they're all currently they're immune to something. All these people on the red on the outside here, they're immune to something. And the idea of herd vaccination is the vast majority of people, in the UK government have quoted 60%, but it just kind of means the vast majority of people in an environment are immune to a particular type of pathogen. This person in the inside here, this person highlighted yellow, they're actually not immune. They haven't had the vaccine for it, but the chances of, let's put some more yellow people on the outside. These people here, they might be carrying the disease themselves. However, there's enough people in this environment, in this sort of section here, that means that the disease is not likely to spread across. So they're protected by the fact that other people are vaccinated. And we do this quite often with animals as well. We herd vaccinate animals to prevent the spreading of disease. And if the vast majority of animals have the vaccines, it's unlikely that the pathogen is going to spread. You've also got ring vaccination, which is kind of why I've drawn this on purpose as well. It's the idea is if you can sort of localise an area and you can then cover the people themselves. So this really is a demonstration of ring vaccination, but herd vaccination is the same idea. Is you vaccinate the vast majority of people, so the transmission is much less likely. A couple of key words moving on around vaccination. So you might hear immunisation. You tend to hear more of an Americanised term, but we still use it in England. So vaccination is the actual injecting, so the process of going and getting an injection in your arm, normally, of the pathogen, whereas immunisation is that plus the response to it. So if a person's become immunised, it means that they have then responded and produced the pathogen. You may be vaccinated against something, but not immediately resistant and immune to a bacteria, whereas immunisation is you've responded and you're now immune to something. Okay, so I've got an example here of this kind of really basic process of how a vaccine works. So a task you could do now is if you pause the video, maybe just minimise the video so you can't see, have a go at creating a flow chart with how a vaccine works. I've got a couple of examples here you could use, so have a go at that. Okay, you can sort of check how you got on. So just to sort of summarise, so we normally inject an attenuated or a weakened version of pathogen. Make sure if you get any exam questions that you make it very clear that the vaccine is going to be an attenuated version of it. The body's immune system's going to fight it off, so we're going to produce the antibodies. Really important we use words like specific antibodies to fight off that specific pathogen. The B and T cells are going to fight it off. We're then going to be left with our memory B cells, and then we're going to be able to, if the pathogen comes back again, we're going to be able to rapidly produce our antibodies again to fight it off, and then we'll end up without being harmed at the end of it. Okay, right, going on to the last kind of section here of the different types of immunity. I will recap in a second. So we've got artificial passive immunity, which isn't as common. So an example is tetanus. So this image of this cat here is trying to show a cat in muscle spasm. I'm sure it's not. It's the best image I could find. Tetanus can cause muscle spasm. Now the problem with that is, if you think of muscle spasm, for the vast majority of the muscles, it's not fatal. However, if that's muscles of the respiratory system, for instance, or the ability to swallow so we don't choke on our own phlegm, it's going to cause us to die pretty quickly. So one of the things that we can do is we can actively inject these antibodies into a person that have already been produced. And we often produce some things like bacteria, and we can inject them or antitoxins directly into an animal or a living organism, and then prevent them from dying from it. And quite often we get some of them from horses, which is why I've left this image here. Okay, so here's a little summary of the different types. It might be worth pausing at this point, seeing sort of what you know, see if you can say each one off by heart, and then perhaps have a go at drawing out a little flow chart similar to mine. Okay, so I thought I'd get some exam questions now. We're going to do some exam questions and a little bit more, and then finish off with a couple more exam questions for this lesson. So, first question. I'm not going to read them out and let you pause the video, have a go, and then I'll go through the answer. Okay, so the answer will be B. So we've got antibodies in mother milk. Remember, it's passive immunity. But however, it comes from the mother, so we will still refer this as being natural. Next one. Okay, so, which is correct, so this is what we talked about, the difference between immunisation and vaccination. So vaccination is the injection of the material itself, and immunisation is actually developing the immunity. Next question. Okay, so, person bitten by a venomous snake, you inject the person with appropriate antibodies, so this is going to be artificial because it's man-made, and it's going to be passive because your body's not producing the antibodies again. Okay, and then the final question of this section. Okay, so why are they active immunity? This question is quite interesting, and I wanted to kind of highlight a couple of things here. First of all, it says explain, so we're not just stating. And we're just looking at active. Okay, we're not looking at whether it's natural or if it's passive. It's just referring to active. And you need to explain it, so you need to make the point, and then you need to give the explanation of that point. So the fact that it's active, it means your body is having to respond. So how do we explain that? Well, we say that antibodies are produced. And then explain, well, how are they being produced? Well, they're being produced by specific memory cells, or you're having to respond to it if it's not a vaccination, if it's just being affected for the first time. So the explanation comes from saying where are the antibodies coming from. Okay, so the next section, I made this PowerPoint slide, this one in particular, I decided to keep it into this a few years ago. Quite ironic timing, because we're in a global pandemic now. So one of the worries that we had previously to pandemics, before this current coronavirus COVID-19 has come around, is that we're worried that we transport so quickly, we've never had this in human history before, around the globe, that instantly millions of people every day are travelling from country to country, and this highly transmissible COVID-19 means that we're in a dangerous pandemic. So, before, we can kind of look at what the government is doing now, but beforehand, our kind of general way that we deal with, and this is just an epidemic, not a pandemic, is we mass vaccinate. So examples of things like Ebola, we've tried to develop vaccines in previous years, and the Zika virus. So we mass vaccinate and then we isolate, which is kind of what we're doing now. We can't mass vaccinate, and then you prevent travel from the infected point, which kind of makes sense. So when this happened in China, Wuhan, people were kind of kept in around the Wuhan area. However, inevitably, it's never going to last completely. And this would kind of be similar with pandemics, but it's more on a global scale. I put this on here, just quite an interesting film about the transmission of a virus within a haunted house, which I quite liked in regards to many kind of these zombie style films. This one has good links to a disease, rabies, which is quite an interesting one to research if you're looking at that. There you go. So let's move on. So let's looking at managing it now with medicines. We're going to go really mostly into antibiotics. So medicines are used to treat diseases, and what we can now build up is these computer models. So here's an example of tRNA synthetase, which is an enzyme which adds amino acids onto tRNA. And we can now use bioinformatics and computer models, and we can kind of develop medicines using computer models. But before that, Alexander Fleming in 1928 came up with penicillin, but we're much more sort of advanced on that now. So what I've done is summarise some common ones. These are the ones from the OCR textbook. But because we've used so much, I'm going to talk about this more in a minute, so much antibiotics now, there's so many that are resistant, so it's really important that we continue to find new antibiotics. Without these antibiotics, we're going to struggle in the future, and we're worried about this huge antibiotic resistance that could happen, because it's already starting. We have this bacteria MRSA, which I'll talk about more in a minute, which is really dangerous because it's resistant to methicillin, which is an antibiotic. So one of the things we need to do, and how we can kind of fight back against this resistance, is we preserve biodiversity. One of the ways that we have so many medicines and ability to fight disease around the world is somewhere in the trees, in the plants, there are all these various different drugs that can help us. For example, we have digoxin digitalis, which is a really powerful drug for people that have had heart attacks and have weakened hearts. quite often, and they have atrial fibrillation. It can really help them, and that comes from a very pretty plant called a foxglove. And there's various other ones. So here's some. It might be worth just pausing now and having a little read through and making notes of this. Okay, just another couple of terms which I saw in the textbook that I thought I would explain for you a little bit. So pharmacogenetics is an example of something that's going on more now, and we're kind of going into this, and this image is trying to represent that. It's the idea of personalising medicines based on a person's genetics. So an example I have here from the textbook is that 30% of breast cancer patients will have a mutation. We call this the HER2 mutation. Now, of those people, we can use specific drugs to help just those 30%, and it's reduced deaths in those patients by up to 50%, which is a huge reduction in those 30%. Whereas the people of the other 70%, this drug doesn't really do anything for. So what we're starting to realise is each person's genome is different, it's unique, and they're going to respond to various different drugs. And kind of an example here, you might have all these patients here with the same diagnosis. Various different responses will be from these people, and we can kind of link that to the genetics. So there might be this kind of group here who are in the best situation where they're going to get a benefit from a drug, and they're not going to find any toxicity from the drug. But then you're going to have kind of various other people, the people on the outside here are not getting very much benefit, and these are even getting a toxic response to the drug. And then synthetic biology is kind of the use of bacteria to produce these drugs for us, or nanotechnology. And you've probably heard of nano before, being very small. So it's the idea of being able to deliver drugs to specific places within cells. Perhaps if cells or parts of cells are toxic to particular drugs, that we can deliver it to specific places, it can then reduce that as well. Okay, so going back to this antibiotic dilemma now then. So we know that antibiotics kill bacteria selectively, so they often work on things like cell walls that us animals don't have. So they're really useful for us because they don't cause any problems in us. In 2014, 1 in 6 prescriptions were for antibiotics, which is huge amounts, and there's huge amounts of over-farming, quoted in places like the US and China, that they pump their animals in farming full of antibiotics, and this is leading to accelerated natural selection against the antibiotic, which means that the bacteria themselves become resistant much quicker than they would normally have, and we're worried that this accelerated natural selection means that we're going to have lots of many resistant bacteria. So 2 examples that I suggest you go away and learn these. So MRSA, which stands for methicillin resistant Staphylococcus aureus, and then you've got this second one here as well, Clostridium difficile, which is quite a common one as well. And they're actually found in the body all of the time, but if they get into particular high levels, they can be dangerous. So for instance, you've got this C. difficile, which is actually going to be in your gut most of the time. However, or in about 5% of the population, they're going to have this. However, if you take too much antibiotics, you can kill off the helpful bacteria inside your gut, which allows then this harmful bacteria to outcompete and then get into high levels that becomes then really dangerous and toxic to us. And then we've got this example of MRSA. Okay, so I've come across this graph which I thought was quite interesting. So this is a very crude, so that means a not very accurate representation of death rate per year, and this is per 100,000 in the US over 1900 to 1996, and we can see where these various events that are going on. We've got this introduction of health departments. At this point here, we're able to now massively start reducing the trend. Okay, at C here we had an influenza pandemic, which was quite dangerous. However, what I thought would be quite interesting is if we were to continue this graph now, I wonder what would happen now. Now we're in a pandemic, the likelihood is we're probably going to see something like that. How high that goes, I don't know. But there's a clear trend here that as we've increased modern medicine, this has started to go down. Okay, right, so that sort of finishes off what I'm going to do for the learning part of this lesson. So I'm going to go over this quite quickly now, so feel free to pause. There's 3 questions. I'm not going to explain them now, but feel free to email me if you want any answers from that. Okay, so question 1, good luck. And the answer here, very important that we talk about specific shape. And question 2, again, feel free to pause the video, nice 6 marker. Okay, so a little bit more difficult this one, so I'll try and give you a little bit of guidance here. So explain why the toxin produced by the different strains. A couple of key points here in this question is you've got different strains. Oops, sorry, I'll try and get my highlight. Explain why the different strains are distinct. Again, what biology likes to do is they like to introduce these terms that they don't necessarily clarify beforehand. So immunologically, so the immune system distinct, separate, will be dealt with. How will these different strains be dealt with by the immune system? So what you kind of have to do here is, if you want to get the higher marks, although you've put lots of things over here, you need to say a description of more than one action of the immune system. So for instance, you could be talking at the antibodies in different ways. Okay, right, I'll let you go ahead and mark that. Okay guys, thanks very much. Just some feedback. If you want me to do things differently, more exam questions, less, faster, slower, etc., feel free to email me. This section was out of, we've got 6, 9, so can you email me your score out of 9, or feel free to take a picture and submit on Show My Homework. Thanks very much guys. I won't release any of the weekend, I'll let you have a weekend off, and I'll be back again on Monday. Thank you.